You must have observed differences in hair color, eye shape, and other traits present in your classmates. Do all your classmates have the same color of hair or eyes, or do they differ in these and other traits? Now, compare yourself with your siblings for eye color. Is there any difference? Humans differ from each other in these and other features, right? We know that eye color, hair color, and many other features of humans are also regulated by genes. We know that genes have alternative forms called alleles. Different alleles of a gene impart different forms of trait to organisms. So, various eye colors present among humans are also the result of different alleles of the gene for eye color. Now, you might ask how these different alleles are produced, which then impart different forms of a particular trait to humans. In other words, what process is responsible for the presence of variations of genetic traits among the organisms? The answer is mutation. In this video, we will discuss mutation and its different types. So let us begin the journey together. Before discussing mutations, let's have a quick recall about the structure of DNA. A DNA molecule has two polynucleotide strands that are composed of 4 types of deoxyribonucleotides. The sequence of these nucleotides in the DNA strand stores the genetic information. But how? The nucleotide sequence of the DNA strand determines the amino acid sequence of a polypeptide chain during the process of protein synthesis. We will not discuss the process of protein synthesis here. However, to understand mutations and their impacts, we need to know that a segment of DNA that specifies the amino acid sequence of a polypeptide chain is called a gene. So, a long DNA strand has many genes that codes for amino acid sequence of the proteins. And the proteins with different amino acid sequence impart different genetic traits to the organism. Now let's resume our discussions about mutations. Mutation is any random change in the DNA sequences of organism. So, any change in the nucleotide sequence of DNA would alter the genes of an organism. Since genes regulate the phenotype, mutations alter both the genotype and phenotype of the organism. You might ask a question here. If mutations impart new phenotypes, are all these new phenotypes beneficial for organisms? In other words, are all variations produced by mutations good for organisms? The answer is no. Changes in DNA caused by mutations are random. So, new proteins or new traits produced by mutated DNA can be beneficial, harmful, or neutral for an organism. Well, I have a doubt here. Mutations change the nucleotide sequence of genes, but is there any effect of mutations on chromosomes as well? Yes, a DNA double helix is packed in one chromatid of a chromosome. So, any change in the DNA sequence also affects the structure and function of chromosomes. Based on their effects on genes and chromosomes, mutations are categorized into 2 main classes called chromosomal mutations or chromosomal aberrations and point mutations. Let's discuss the chromosomal aberrations first. As the name suggests, the chromosomal aberrations are change in structure or the number of chromosomes caused by mutations. Let's have a quick look at different types of chromosomal mutations. First is deletion. The deletion occurs when a part of a chromosome is missing, making the chromosome deficient in an amount of genetic material. In the given chromosome, the segment labeled A is missing due to deletion. When a part of a chromosome is repeated more than once in a chromosome, it is called duplication. The segment AB is present twice in the duplicated chromosome. Now, in the given image, the segment ABC from this chromosome is inverted in the form of CBA in the inverted chromosome. Change in the direction of a chromosomal segment is called inversion. The segment B from chromosome A has been added to chromosome B. So, chromosome B has undergone insertion mutation here. So, what is insertion? It is the addition of genetic material to a chromosome. Here, the chromosomes A and B exhibits an exchange of genetic segments and results in translocated A and B. Chromosomes A and B have genetic material from chromosomes B and A respectively. When one chromosomal segment is attached to a different chromosome or a different part of the same chromosome, it is called translocation. Let's discuss the other category of mutations, which is point mutations. Point mutations are the mutations caused by a change in the single base pair of a DNA molecule. Let's take the example of a disease caused by point mutation. Red blood cells have a protein which is called hemoglobin. Hemoglobin proteins have alpha and beta chains respectively. When single base thymine from the beta globin gene of hemoglobin protein is replaced by adenine, the triplet codon GAG is altered to GTG. GAG codes for glutamic acid while GTG codes for valine. This change in the single base pair in the gene is called point mutation and causes the disease sickle cell anemia. Here, we see that a mutation causes a disease. You might ask a question here. What causes the gene for the beta globin gene to undergo mutation, which in turn results in a disease? Do this point mutation and other mutations occur naturally? Are there any substances that induce mutations? Spontaneous mutations occur naturally, while induced mutations are caused by specific substances or factors. These factors that can induce mutations are called mutagens. Mutagens include both chemical as well as physical factors. Let's take some examples of each of these categories. Nitrous acid is a chemical mutagen that removes the amino group from the nitrogenous bases. Nitrogen mustard and ethyl methane sulfonate add alkyl group to the nitrogenous bases. So, nitrous acid, nitrogen mustard and ethyl methane sulfonate are chemical mutagens. X-rays delete bases, introduces single-stranded cuts in DNA and break in chromosomes. UV light triggers the formation of dimers of pyrimidine bases such as thymine dimers. Therefore, we can say that X-rays and UV light are physical mutagens. We have already discussed about sickle cell anemia, which is caused by point mutation. Are there any other disorders caused by point mutations? Do chromosomal aberrations also cause genetic disorders? If yes, these disorders should be inherited from one generation to the next, right? So, can we predict the occurrence of a genetic disorder in progeny if the parents are affected? We will find out the answers to all these questions in the next video. Now, let us summarize what we have learned.
Manorama Horizon. Learning made simple.